Thank you. So I'm Estelle Vegan, and I'm here to tell you the story around the successful development of high quality lipids and why this is critical now for the development of RNA delivery systems and vaccines. And so let's get started with the first dimension across our name over different regions of the world. And so now onto the topic of RNA. As API, one thing that is important to look back a little bit into our high school biology as to why those RNA and DNA can actually be used as API. And this comes to the fact that diseases often manifest themselves through the alteration of protein expression. And as DNA and, for example, mRNA can modulate protein expression, this is why they can be used as API. A few differences, though, that should be highlighted between those is that, as indicated on the picture here, you have DNA that would need to be within the nucleus in order to act, compared to mRNA, for example, which can act within the cytoplasm of the cell. And so this is one of the components that would be definitely very different in terms of drug delivery aspect that's important to consider. Another thing that is also very much needed to notice is in terms of the DNA, in order for it to act, and you have this integration as part of the genome, that is something to consider that is not required as part of, for example, the delivery of mRNA. So in that sense, you have a little bit of that safety measure with the mRNA that only acts in this very transient nature for the protein production. So now that we've looked a little bit into this background as to how RNA can be a drug, one thing that we can look at is now what type of indications it can look at. And so here I'm focusing on all sorts of RNAs, so looking at small RNAs as well as long RNAs. These can, in general, lead to the production of antigen and protein, for example. It can also act as a gene silencing or activation pathway. It can also be used for enzyme replacement therapy, for example, and therapeutic antibodies, as well as gene editing and with CRISPR-Cas9, for example. And all of these, then you can see that this is quite a broad range already. And really this kind of looks at this wider range of indication, from vaccine to cancers to pulmonary diseases, as well as liver metabolic disorders, for example, and precision medicine. So you can really see that this is a broad range of targetable indications, which really highlights the large potential of RNA as API. Now, though, if we look at specifically in the case of COVID-19, for example, and so you have a few different pathways in which RNA can act against COVID-19, for example. Here, as depicted, you have in the sense of mRNA for the antigen to enable this antigen delivery, and you can also have the mRNA for antibodies, and so you can focus more specifically on the antibodies, as well as if you look at RNA interference, for example, this is when you can stop the viral replication. And so these are a few different pathways that are possible, and that can be looked at in the specific case of COVID-19. Now, if we look at the pipeline of RNA projects that are currently ongoing through the time of COVID-19, so you have February last year all the way to June this year, what you can see is that really the RNA projects that have been recorded have increased a lot over this time period. And if this is segmented over the different delivery methods you have here through the different colors, what you can see is that lipid is one of the delivery methods that is the most significant for this RNA pipeline. And so if we look at these lipids more specifically, they have a few different ways that they can be assembled to generate a drug delivery system, and that can be liposome, lipoplexes, and lipid nanoparticles, also called LNPs. And the reason why these are versatile drug product is because they are a little bit of this plug and play system because of the self-assembly of these lipids. They can enable the co-delivery of multiple API, for example, multiple RNAs or RNAs combined with small drug molecules. So this is something that can also be integrated. So they add this flexibility that really, I think, is indicated here with this overview of the pipeline. You can see that lipids are used for the delivery of RNA projects. Another component that is highlighted here is the immunostimulation aspect of lipids. So some of the lipids, depending on which ones you use in your formulation, can have this characteristics which can be advantageous for your project if you're looking for an immunostimulation response. So now, one of the main things that is affecting the performance of the formulation of those LNPs is the composition of your formulation. And so you have here bucketed into these 4 groups the different categories of lipids that would generally be included in your formulation of lipid nanoparticles. And so the first one would be cationic ionizable lipids, which enable the encapsulation and the release of your nucleic acid cargo. And this also something to note has an influence over the toxicity of your formulation, and so this is something that is to be considered as well as you formulate your systems. The second group would then be neutral lipids. Those are also called helper lipids. So these act as enabling or enhancing the structure of your lipid nanoparticles. They can also add fusogenicity property to the system and to the formulation. And in general, these would be reducing the toxicity of your formulation. So these are can work hand in hand with the cationic ionizable lipids. The third group would be pegylated lipids, which are used for stability enhancement. So you had, for example, if you consider group 2 with the neutral lipids, and you have the structure here that also affects the stability of your LNPs. But when I'm talking about the stability on the sense of pegylated lipids, this is more focusing on the circulation and the stability of the particle as it's administered so that it doesn't aggregate and such. So this is one of the criteria why pegylated lipids would be used. Depending on which pegylated lipids would be chosen for the formulation, then they can also enable or can have an effect on the release of the RNA. And also known as well for the pegylated lipids is their potential immunogenicity. And I'll touch a little bit on this in the coming slides. The last group here is then cholesterol, which provides structural integrity to the lipid nanoparticles by filling the gaps between the tails of the lipids that are used within the formulation. And this is why cholesterol plays a major role there. So some of the consideration, obviously, is which lipids should we use, in what ratio should we have the lipid versus the RNA payload that you're trying to encapsulate, what's the administration route that we would be focusing on. And so these are all considerations that are important to define as you get into formulating your LNPs. Some of the how to, I guess, go about answering those considerations. So which cationic ionizable lipids should we be using? This is goes back to your definition of what potency and what targetability and immune response you're trying to achieve. And this is an important question to answer for your formulation. For the neutral lipids, so for the helper lipids, in terms of choosing that, this depends on what your definition of what stability you're trying to achieve with your particles. And same for the pegylated lipids. So it would be important to define at that point the optimal length of the carbon chain as well as the peg that is required for your system. And one component as well, very relevant for cholesterol here, is the lipid that you're using, animal-derived or synthetic, and this is relevant for cholesterol, as I'll touch on in the coming slides as well. So now we will do a deep dive into each one of those buckets. And so the first one, cationic lipids, I will focus on the example of dotachloride or as well as DOTMA. So you have both of them depicted here with their structures. And so really these cationic lipids would be through electrostatic interaction encapsulating, enabling the encapsulation of the nucleic acid or the RNA within the liposome and the LNPs. And so this is how it would enable the encapsulation of these systems. It can also promote the release of the payload. And one important consideration to have as well is that most or all LNPs contain cationic ionizable lipids, and generally this is a majority of the overall formulation. So choosing that cationic lipids in terms of, to achieve the performance that you're looking for is definitely critical. Then in terms of considerations, the critical raw materials for this is very much one of the components within your formulation that will affect the performance of your drug product. And so we've seen also that this is a space for innovation, of course, and definitely if you have proprietary lipids that you would like to be using in your formulation, then as you get to commercializing your product, finding a CMO capabilities that follow you through that process and scales with your need is definitely something that is critical. Another factor that is important to note for the cationic lipids is the requirement also for endotoxin testing. That might not always be feasible for cationic lipids, depending if you're working with a strong cationic lipid or not. And this is something that to be considered as endotoxin testing becomes a requirement for these drug delivery systems. This is something to be considered as it's not always feasible, and the protocol for endotoxin testing might then be need some adjustment in order to enable this. Now, the next bucket would be looking at the neutral lipids. So these are the helper lipids that enable the stability of the structural stability of the LNPs. So here I've put the example of DSPC, DOPC, and DOPE. So you have DSPC with a stable laminar phase and a high melting transition temperature with 55 degrees Celsius. And this is very much in contrast to DOPC, which is in a fluid state at physiological temperature with a low melting temperature. And this is also contrasting with the OPE, which has a hexagonal phase at low pH, therefore can then, in some cases, promote the endosomal release of the nucleic acid, which is something that is very much needed also in some of the formulations. So this is why these would be considered as neutral lipids for the formulation of RNA particles. Then in terms of considerations for these ones, obviously, the stability, the structure, and diffusogenicity properties that you're trying to achieve will very much guide your selection for that neutral lipid. And again, these neutral lipids, they're very much in your toolbox in terms of enabling the characteristics that you are looking for within your formulation. Then moving on to the pegylated lipids. Here I took the examples of DMG and DSG-PEG. You have them depicted here on the right with these two. And so you have DMG with a shorter anchor length of C14 compared to DSG with C18. And so the dissociation time of this lipid from the formulation will then be faster for DMG compared to DSG because of that shorter anchor length. And so, yeah, again, this dissociation of the PEG lipid from the formulation as it's administered will then have an effect on the transfection and the gene expression ultimately. So this is something that needs to be defined and needs to be tested as part of your formulation to understand which one will basically enhance the transfection and the gene expression that you are looking for. PEGs, of course, shield the particles from the immune clearance and prevent aggregation and increase circulation time, which is a topic that I touched on earlier. and is the reason why we need to include those within the formulation. However, PEG lipids are in general in small proportion compared to the rest of the formulation. However, but they're still necessary in order to improve the stability of the system. We've definitely very much seen that there's an increased research right now for PEG alternatives, as we are looking for non-immunogenic solution to this with similar properties. And so this is very much an area in focus right now, and something to keep in mind as you consider the need for PEG lipids within your formulation. Then the last bucket here would then be cholesterol. This is very much an essential part of these formulations. It again provides structural support and rigidity to the particle. And the majority of liposomal drug products contain cholesterol, and all of MNPs do. As you have also the little bit of schematic here that shows basically the marketed liposomal drugs considered, the majority of them do contain cholesterol. Some of the considerations that come around cholesterol and the topic of cholesterol, that goes around the fact that cholesterol, we've seen recently with the increased demand for lipids through COVID-19 and through the pandemic, this is an essential part of the formulation and is very much needed across the board. And this is when having a supplier with consistent supply chain and flexibility in manufacturing scale is definitely, these are crucial in order for you to have a consistent supply of this material for your formulation. And so this is an important consideration, definitely. In terms of synthetic material versus animal-derived product, this is something that I wanted to highlight as well. Synthetic versions of cholesterol, they enable really that low impurity profile that is very well defined and consistent batch to batch, and definitely come from a safe source because it's synthetic. And therefore, there's no quantity restriction around the use of that material. And this is in contrast to the animal-derived counterparts, which would have restrictions. And so this is important to consider as well as you formulate your products. Now, if we look a little bit around the definition of the quality of lipid, we say it is important, but it's important to also understand why it is actually. Why do we highlight it so much, and why it's crucial really for deformulation? This goes around really the topic of that's highlighted on the left-hand side, and this is when you have, you can have high purity, consistency, and good material characteristics when you go for quality lipids. High purity really highlights the fact that these lipids will have good stability, and therefore your drug products will have good stability. It will have expected drug release profile as highlighted here, and in terms of consistency, these are going for reproducible results, therefore avoiding the need for repeated studies in order to justify your formulation or your results. And in terms of material characteristics, this goes in the sense that, having good quality lipids then leads to good material characteristics such as good solubility, stability, as I've highlighted, good flowability and handling properties, so that then as you scale up, then the material can continue to follow you and you can continue using the same material as your scale increases. And this is something that's very much needed as, to expedite any of these timelines. As we know, all these timelines are very much expedited nowadays. And so quality overall, this implies expected results, no regulatory hurdles, cost saving because you don't have to repeat some of these studies, and overall an ease for these drug product manufacturing and for you. Some examples right now that are exactly relevant to this topic of quality that are highlighted here. So we have DOPE that you can find in wax-like form. So this forms kind of lumps and gels. And obviously, when it comes in this form, this is when you have issues with solubilities, even if the supplier says that the product is lyophilized, for example. And this is when, through process development, we've been able to generate a product that is in powder form. It's free-flowing powder that really that is easy to handle and enable really that ease of manufacturing thereafter, and also has very good dissolution properties compared to the wax-like product. And so this is definitely something to highlight here. Similarly, for DOPC, we can start with an amorphous DOPC, as highlighted on the left, and through process development and that know-how that we have for our lipids, we've been able to generate crystalline DOPC that you have highlighted on the right, with good handling properties because of that powder form. And therefore, it also has enhanced stability compared to the amorphous compound and faster dissolution rate, which are important criteria for this material. Then we will get a little bit into the topic of the analytical tests that are required for these lipid drug delivery systems. So you have first the physical characteristics, such as the size and the polydispersity index. You would also have a requirement around characterizing the pH, the zeta potential, the transition temperature of your formulation, and the lipids as well. And also, so you have all these different analytical tests that are required for your formulations. This also then applies to the chemical characteristics, where identifying and quantifying actually the drug product compounds and their degradation is a requirement. Similarly, same for lipids, they need to be identified, and the impurities also, as well as quantifying them and knowing knowing in what quantities they appear. Same for residual solvents and elemental impurity. These are requirements that are definitely highlighted nowadays, as some of these requirements are becoming stricter and stricter around these drug delivery systems. And in terms of the biological analytical tests that are required, this is when you have the requirement for sterility of the final drug product, and also a need for endotoxin measurement. And so this goes back to my mention earlier about the cationic lipids. This is when this is something that would need to be considered. Then continuing on this topic of the regulatory considerations, lipids used in liposome formulations are highly regulated, and this is also extended to the LNP, to the lipid nanoparticles formulations. And one of the critical questions to answer is around if the lipid that you're trying to use is an excipient or a novel excipient. And we've provided a little bit of a workflow around this to try to answer this question. Is basically, when you start with your lipid, does it come with a monograph? Does it have a monograph? If it does have a monograph, then that's a fairly straightforward path. You refer to that compendium, and that's straightforward. Then if it does not have a monograph, then you have a few follow-up questions. Then the first one would be, is it a new entity? If it is a new entity, it would automatically be categorized as a novel excipient. If it is not a new entity and it's already then the follow-up question would be, has it already been used in human applications? If it has not been used in human application, it again goes back to being categorized as novel excipient. If it has been used in human application, then the last follow-up question would be, was it administered in human application with similar dose and similar route of administration? If it was not similar, then it would be still categorized as novel excipient. If it has, then the cross-referencing pathway of the previously used excipient is something that is feasible. And so this is something, this workflow is definitely important to consider as you're looking at different lipids for your formulation. A few points here, different across the world. So basically you have the European Union with the excipients that are only filed as part of the marketing drug application. And there's also no specific guidelines around novel excipients or lipids as excipient, to be specific here. And so this is something that's important to know as well. And this is in contrast a little bit to the US with the FDA and the implementation of the excipient master file, so the EMF, which is then a type 4 DMF for excipient. And one thing to note here is, in addition to other requirements, there's definitely a need around quality-related data, such as a tox study, for example, and stability data around the lipids that are used and the excipients that are used. However, in the US, you do have that specific guidance that I've referenced in the previous slide. So you have specific guidance around liposomal drug products that's also used in reference for the lipid nanoparticles. So overall, this is an extensive process. It is definitely complicated, and there are learnings around it. There's limited guidance, for example, right now around a novel excipient filing within the EU. And this is when really choosing a supplier that has experience in lipids regulatory filing is key in order to avoid any hurdles around this topic. Then this is when we go on to choosing a partner, and this is when it becomes very important. The characteristics of the right partner, it goes around knowing that your partner uses high quality products and provides high quality products themselves. This goes on to the topic of quality that I mentioned earlier, as well as services. So we need high quality, but also consistency in the GMP raw material that we would be providing. A deep know-how within the topic, within the products that they're supplying, a deep know-how within the raw material that they're using, so that they understand how to expedite any of these timelines for the manufacturing of these large-scale GMP excipients, as well as I've just mentioned, the regulatory landscape, which is complex, and so experience in that field can really prevent some of the hurdles around the regulatory filing. Right facilities, this is definitely highlighted as we've had this recent surge in lipid demand. This is when you need to choose a partner that has this flexibility in manufacturing scale and can therefore safely provide a consistent supply of material to your projects. And so this comes to the topic of how can we support lipid-based drug delivery. And so our offering would definitely be focused a little bit on custom manufacturing as well as our lipids portfolio. So we have both of these capabilities. For example, if you have proprietary lipids in terms of your ionizable cationic lipids or pegylated lipids or any of those, this is something that would be considered a special lipids, which can be custom manufactured through the different scales that you need. And in terms of our lipids portfolio, this is when we're focusing on providing high quality synthetic lipids with a broad range of products to select from. But there's some customization that is feasible even within our portfolio. For example, if you require specific pack sizes for your manufacturing, this is something that is also feasible. Then in summary, quality definitely leads to performance of your drug product. And so there's a plug and play nature around the formulation of lipid drug delivery system. It's important to plan ahead and look at where you're trying to go with your formulation. If this is a timeline that needs to be expedited, having an intelligent formulation design is definitely important to expedite any of these timelines. In order to ensure quality, this is something that is very much highlighted throughout this presentation and was a theme across in order to have reproducible results and again, just avoid wasting any of the materials and wasting resources as any of these timelines where are very critical. And this when the last point here is choosing the right partners. As you choose the right partner, it can really help in terms of avoiding any of the hurdles, as I've mentioned, for example, in terms of regulatory filing. And this was kind of my last slide. So if there are any questions, I'm happy to take them.